Good morning all. Today I am taking a lecture on corynebacterium. Okay, corynebacterium is a genus that contain the different species, that is diphtheriae and some other else, that is called the diphtherioids. Okay, so diphtherioids are the saprophytes, but the corynebacterium diphtheriae is a causative agent of diphtheria. So first I want to give the introduction about the corynebacterium diphtheriae. They are the gram-positive, non-acid fast, non-motile rods with irregularly stained segmental and sometimes granulars. They shows frequently club-shaped swelling, and that's why its name is called as corynebacterium. That means coryna means club shape. The diphtherioids and the coryniforms have been used to describe other corynebacteria that do not commonly cause human infections and were considered less significant. The major disease that is caused by the corynebacterium diphtheriae is a diphtheria. The today's lecture we want to discuss uh, like classification, morphology, and some toxic substances which is produced by the corynebacterium. So first we'll discuss about the classification. So the corynebacterium diphtheriae can be classified as a saprophyte and deceased one. Generally they produce three type of disease, that is called uh, clostridium gravis, emmet and intermediate type of disease, in which the gravis type is highly pathogenic. Okay, so I'll start the discussion with morphology of corynebacterium diphtheria. After that we will discuss the classification. So it's a small gram positive uh, bacilli, and their arrangement is like a palisade or the Chinese letter. Have you seen the Doraemon letters like that? The Chinese letter arrangement is there, and generally they have a metachromatic granules also, by which they appears the black is color at the ending. Okay, so if totally I want to totally if we want to discuss, so we can say that they are thin, cylinder, gram positive bacilli, but easily decolorize, and particularly in the old culture medium its size is different, like 3 to 6 micrometers. Okay, and its width is about the zero point 0.6 to zero point 0.8 mu. They have a tendency to clubbing at one ends, or it can be of the two both ends. Okay, and sometimes it will shows the pleomorphic activities also. The bacilli are arranged in the characteristic fashion, that is called the Chinese letter arrangement or the cuneiform arrangement, and they are usually seen in the pairs and sometimes in the small group also. And this arrangement is due to the incomplete separation of the daughter cells after the binary fission of the bacteria. So this bacteria that also have some granular like structure, and some uneven they show some uneven staining also when they stain with a methylene blue or toluidine blue. The granules in the cell stain metachromatically reddish purple. These granules are known as metachromatic granules, polyethylene granules, or Babes Eastern granules. They are often situated at the poles of bacilli and are called as polar bodies. Okay, special stain are used for the staining of these bacilli. That is called as Albert staining or Neisser staining or Pond staining. And by using of this kind of technique, we can easily distinguish between the granules and the bacilli. Okay, so when we stain with Albert stain, the granule stain will be of the bluish black in color, and protoplasm remains green in color. The granules represent the accumulation of the polymerized phosphate. That granules formation is best seen in the Loeffler serum slopes, and it seems that they represent the storage deposits for the material needs to form a high energy phosphate bodies. These organisms are non-motile, non-sporing, and not non-acid fast. Okay. Um, on the basis of classification, some they are the saprophytes, some produce the disease in the animal, and only the one C. diphtheria is a causative agent of this. You can see that these are the this diagram is showing the pleomorphism of diphtheria, and strain is of gram strain. As we discuss here that they have a palisade arrangement or Chinese letter arrangement, see that this type of arrangement can be seen in the staining. Now, culture characteristics. The the screen showing that the Loeffler serum slope, it is a specific media for the growing of growing of uh, corynebacterium diphtheriae, on which the colonies can appear within a 6 to 8 hours. Okay, but in the normally all the features in the culture characteristics are they are aerobic and facultative anaerobes. The optimum temperature for the growth is 37, and it could be the range up to the 15 to 40 degree, and the optimum pH is 7.2. The complex media is required for the primary isolation and characterization. It can grow on the ordinary nutrient media, but its growth is improved by the presence of animal protein such as blood or the serum. Two media are useful for the purpose, that is of Loeffler serum slope and blood agar, containing the fresh, lysed and heated blood. Okay. We can also say that C. diphtheriae is a fastidious organism. They are best grow in the enriched media, not in the normal media. When we take this sample from the Loeffler serum slant, so the methylene blue stain, if we stain the bacteria with methylene blue stain, we can see this type of arrangement. Okay, Loeffler serum slope. Diphtheria bacilli grow on the Loeffler serum slope very rapidly. Uh, Within 6 to 8 hours, colonies produce long before bacteria grow. Colonies are at first very small in circular and opaque disc-like structure, but they enlarge on the continuous incubation and may acquire the distinguished yellow tint. Okay. And next is called as telluride blood agar. Telluride blood agar is a selective media for the growing of corynebacterium diphtheriae. On this media, only selectively corynebacterium diphtheriae organism will grow, and other organism is restricted. Okay. So the addition of the potassium telluride, that is 0.03 to 0.04%, it makes the medium selective for the corynebacteria by inhibiting most other pathogenic and commensal bacteria on this medium. C. diphtheriae gives the grey, black, shiny or dull black colonies because the telluride ion pass through the cell wall and membrane into the cytoplasm, where it reduces the metal tellurium and precipitate inside the cell. Some other corynebacteria like Staphylococci and the yeast reduce the telluride salt, producing the characteristic grey to black colonies. The addition of cysteine to the telluride containing media, that is called as Tinsdale medium, has greatly helped the isolates of diphtheria bacilli. The growth of the diphtheria bacilli may be delayed on the telluride medium, and the colonies may take 2 days to appear. Based on the colony morphology on the telluride medium and other properties, MacLeod has given the classification that is called Gravis, Intermediate and Mitis type of classification. And the names were originally proposed to relate the clinical severity of the disease produced by the three types. Gravis cause the most serious metis and metis, and the mildest variety, with intermediate being responsible for the disease of intermediate severity. Okay, and in general, biotypes with the predominant in the endemic area, while the intermediate and gravis tends to be epidemic. So, um, if we are discussing about the culture characteristics, two important media, Loeffler serum soap and telluride medium. On the basis of telluride medium, that can be classified the three type of species. We are discussing here the three type of species like gravis, intermediate, and metis. Uh, First, I want to take the morphology. So usually gravis type of stain, they will produce the short rod uniform staining, while the intermediate they will produce the long and cuboid type of morphology. Sorry. And the metis, they produce the long and curved rods. Colonies on the telluride blood agar, they will, after the for 18 hours of the growth, the colonies will appear 1 to 2 millimeter in size. They have a grayish black color center, and within 2 or 3 days 3 to 5 millimeter in size. Flat colonies with raised dark center and centrally edged with radiation stration, that is called the daisy head colonies. After the 18 hours of the incubation, the colonies in case of intermediates is almost about 1 millimeter to 1.5 millimeter in size. Misty type of colony is there. And after 48 hours of incubation, the dull granular central with smoother most glistery periphery colonies can be appears. This type of appearance is called as frog's egg colony. Metis type of infection, that is of size variable and shiny black, almost 2 to 3 days colonies become flat with central elevation. Poaches egg colonies like appearance is there. Consistency of colony if we want to discuss, so gravis will produce the brittle type of colony, intermediates will produce the normal and mediocre type of colony, while smooth and buttery colonies will be produced by the metis type of strain. Hemolysis, that is variable in the gravis, non-hemolytic in the intermediates, and usually hemolytic in the metis type. And toxic strains. Toxic strains are almost 100% toxic strains are of gravis. 95 to 95% they are of intermediate type, and 80 to 85% they are of metis type. Okay. If we want to discuss about the virulence is great is present in the gravis, and intermediate that is of moderate type. Metis, they are of the mild type. Predominant strains in the epidemic areas, okay, and intermediates is the epidemic areas, and metis in the endemic areas. So this type of classification is based on the clinical severity. And we have discussed here about the uh, corynebacterium telluride medium. On the basis of this, we have shown you the classification. And corynebacterium gram positive organism that inhibits the gram negative on the uh, potassium telluride medium. Cysteine telluride has longer shelf life, and Tansdale helps to differentiate among the corynebacteria. Okay, colonies on the either appear black or the grey due to the telluride reduction. Staph aureus and Listeria also represent black colonies, and Tansdale, C. diphtheriae, ulcerations, and pseudotuberculosis form the brown halos around the colonies due to the formation of ferric sulfate. Okay. Okay. In detail, we have discussed about the three morphological type of C. diphtheria on the basis of clinical severity. Metis, black colonies with grey periphery. In short, we can discuss the gravis, large grey colonies, intermediates, small dull grey colonies. All produce the immunological identical toxins, but the Severity is different according to the strain, and incubation period is 35 to 37, and preferably pH is a 7.8 to 8.0 for the good growth, and it requires the access of oxygen, that means poor ANO2 growth. Okay, because airways पे present है, so that is very much aerobic. Okay, next is a biochemical reactions. About the biochemical reactions, corynebacterium diphtheriae, that ferments the glucose and maltose with production of acid, but gas is not formed, and in Durham's tube there will be no gas, and bubble will be shown by the bacteria. But non-lactose, mannitol, tetralactose or sucrose, that is not fermented by this bacteria. Starch and the glycogen are used for the biochemical identification of the three biostrains between the gravis strain and metis and intermediate type. Um, the gravis strains, they will produce the positive strains of the starch, and intermediates that will produce the negative reactions, while the metis strains are also negative. Okay. So fermentation of the sugars are usually done in the his serum peptone water medium, and C. diphtheriae, that is in H2S positive, and reduce the nitrate to nitrites. Okay, it does not liquefy the gelatin or hydrolyze the urea or form a phosphatase. Okay. Mm. One test is called as uh, parazinamide, that is PY is a test. This test is helpful for distinguish between the C. diphtheriae and other form of the C. corynebacterium diphtheriae. uh, Toxins that is produced by the C. diphtheriae. It's very powerful enterotoxin. The toxicity observed in the diphtheria is directly attributed to the toxins secreted by the bacteria at the site of infection. Okay, synthesis almost 95 to 90% strain of the gravis and 80 to 85% of the metis, they will produce the toxins, and that these all toxins will produce the cytological effect. Okay, and that is very much important that they are the catalase positive, they are non-motile, and corynebacterium diphtheriae ulcerine is urease positive, and diphtheria is negative, and C. pseudotuberculosis is also usually positive. Virus factor we have discussed here, and C. diphtheriae also causes the diphtheria, and enterotoxin is there, and heat labile polypeptide lysogenic type of beta phase toxic gene is there, so that will produce the toxins, and pH that is good for the bacteria is 7.8 to 8.0. Aerobic condition is good for that. Low environmental ion levels, that is essential for the toxin production, and toxin inhibits the protein synthesis by ADP, ribosilation elongation factor 2, and same type of toxins is also produced by other bacteria that we will discuss in the further topic. Okay, now uh, properties of the toxins. The diphtheria toxin is a protein that have a molecular weight of 62,000. It consists of two fragments, that is called the A and B. The molecular weight is about 24 to 38. That means molecular weight of A is 24 and B is 38. And both fragments are required for the toxicity, because fragment A has an all enzymatic activity, whereas a fragment B is responsible for the binding of toxin to the cell. The antibody to the fragment B prevents the binding of toxin to the cell, and this protective. The toxin is heat labile. It is extremely potential. That means 0.0001 mg. That will kill the bacteria, guinea pig. Okay, and this toxin can be changed into the toxoid. What are the toxoid? Toxoid are the substance that is formed by the toxin, and that they lose their toxicity and has been used for the production of the antibodies and antigenicity as an antigenicity. The mode of the action is the diphtheria toxin act on the by the inhibiting the protein synthesis. It inhibits the polypeptide chain elongation in the presence of NAD, nicotinamide adenosine dinucleotide. By inactivating elongation factor 2, the inhibition of the protein synthesis is probably responsible for the both the necrotic and non-necrotic effects of the toxins. Okay. Uh, in case of this toxin, the trypsin cleave the toxin into the two fragments, the A and B, and both have a disulfide bridge. B binds to the tissue and A contain the toxicity. The molecule of the toxins can be inhibited 90% and protein synthesis in the cell. Systemic effects include the heart failure, paralysis, adrenal hypofunction, and Addison's disease. That is the most clinical symptoms of this infection. See, Alzheimer's and pseudotuberculosis sometimes make a diphtheria-like toxins. Okay, how they perform their work? The toxin enter through the receptor-mediated endocytosis. After that entry, the acidification of the endocytic vesicles allow A to dissociate from the B. Is A and B both will be there. B is binding, A is attached to the B, and after this enzymatic actions, the A is moved from the apart from the B, and that enter into the cytoplasm. You can see diphtheria toxins. You can see this is a mode of action, how the diphtheria toxins works. To prove that isolate can cause the diphtheria, one must demonstrate the toxin production, like that is called the Alec plate. Before seeing that, just we want to discuss about what is the pathogenicity of this bacteria. The organism is carried out by the upper respiratory tract and spread by one person to another person by the droplet infections or by the hand to mouth touch. Okay. When the organism get enter into the body, the incubation period is about 2 to 5 days, depend on the patient's immunity, and sometimes it may take the 1 to 10 days. Diphtheria, which occurs in the two form, that is called the respiratory and cutaneous form, that is of the worldwide. If we are discussing about the respiratory diphtheria, so the illness begin gradually by the characterization of the low grade fever, malaise, mild sore throat. The most common site of the infection is the tonsils or the pharynx. The organism rapidly multiplies an epithelial cells, and toxigenic strain of the C. diphtheriae produce the toxins, cause the tissue necrosis and exudate formation, triggering an inflammatory reactions. The combination of the cell necrosis and exudate forms a tough gray to the white pseudomembrane, which attach to the tissue, commonly over the tonsils, pharynx or the larynx. Any attempts to remove the pseudomembrane resulting in the bleeding. In case of nasopharyngeal infection, the pseudomembrane may involve the nasal mucosa, the pharyngeal wall and the soft palate. In this form, edema involved and cervical lymph gland may be there. Their involvement is there, and this condition is called as bullneck diphtheria, and laryngeal involvement leads to the obstruction of the larynx and lower airways. If it is untreated at that point, so that will change into the systemic effect. So the toxin also absorbed and can produce a variety of systemic effect, like that can affect the different organ like kidney, heart and nervous system. Uh, in the heart, they will produce the myocarditis, peripheral neuritis and thrombocytopenia. And generally this type of effect, if it is there, so the death is due to the congenital heart failure and cardiac arrhythmia. If it is treated but not properly, So that complication can be of that asphyxia, acute circulatory failure, and post diphtherial paralysis. Sometimes septic infections like pneumonia and otitis media is also can be seen if it is untreated. So the complications are can be there. Okay, second type of diphtheria is called as cutaneous type of diphtheria. In this cutaneous type of diphtheria, in which that is prevalent in the tropical countries, and the toxin also absorbs systematically, or systemic complications are less common than upper respiratory system with C. diphtheria. So mainly the visible effects are generally we can see in the laryngeal and pharyngeal diphtheria, not in the cutaneous type of. For the showing of this uh, toxicity, the Illich gel precipitation test which we are using for the detection of toxicity, that is um, in plate, normal media plate, organism is streak on the plate containing the low ion, a filter strip containing the antitoxic antibodies placed perpendicular to the streak of the organism, and diffusion of the antibody into the medium and secretion of the toxin into the medium occurs. At the zone of equivalence, the precipitation will form. You can see in the diagram that is, you can see that Illich gel precipitation test that is used for the showing of toxicity of bacteria. The line of density, you can see that antitoxin impregnated filter paper, okay, and bacterial growth and a specific peptide line means bacterial growth on which the antitoxin is also there. So where the antigen will combine with its specific antibody, the precipitation of line of precipitation will be marked, and that can be observed in the Illich gel precipitation test. Okay. Next test we are that is called the Illich gel. Now sometime it may be used um, the toxicity test or a virulence test in the animal also. That is called the Schick test, virulence test. So we will take the guinea pig or the tissue culture toxicity assay may also can be done. Capsule that is of protein in nature, and cod factor that is complex glycolipids, okay, that has been shown to disturb the mouse mitochondria. It has also been shown in the play to role in the production of diphtheria. Okay, so in vivo test we will do the subcutaneous test and intracutaneous test. In vitro test we are also using the precipitation, tissue culture, enzyme link immunosorbent assays, and polymerase chain reaction also. For the toxicity we have discussed here about the Schick test, okay, and Illich gel precipitation test. For the prophylaxis we are using immunization, active immunization and passive immunization. Sometime for the treatment the combination of immunization is also required. By the active immunization we are using the DPT, triple vaccines, with a dose of interval of three times as a booster. Passive is used for the given as a 500 to 1000 unit as a treatment like antitoxins, and combined therapy is also useful when the patient is there and if he and she is taking the treatment of diphtheria also. So the for the treatment, the specific treatment for the diphtheria consists of the treatment of the antitoxic substances in simultaneously with antibiotic therapy, and antitoxin, antibiotic, and some other management also. Okay, so for the line of treatment it could be the penicillin and erythromycin, and diphtheria usually start as a local infections We can thought that this is a mild disease, but that could be the dangerous, that may obstruct the airways and result the suffocation. Penicillin which we are using, prevention and active test, Schick and Dick test is used for the detection of the corynebacterium diphtheria and for the immunization. Last, we can discuss about the diphtheroid, that corynebacterium resulting C. diphtheriae. They even is a normal commensal for the throat, skin and other areas. Mainly they are non-pathogenic, but they are living within the cells. Okay. Um, Next is uh, important features like mm, see if there is a confusion between the C. diphtheria and diphtheroid, so we should have an idea about the different tests by which we can differentiate that, because the toxic strains are C. diphtheria, diphtheriads are non-toxic. Virulence tests are the positive in the C. diphtheria and negative in the diphtheroid. And culture, that is grow best grow in the C. diphtheria on the enriched media, and they can grow easily on the ordinary media. Okay, so this is all about the today's lecture. I hope आपको अच्छी तरह समझ आया होगा. Thank you very much for being with me. Thank you very much.